The second experiment that we did in this particular lab was called chromatography. Okay. Chromatography is a technique used to separate the components of a mixture based upon a chemical or physical property. In our lab, we separated pigments based upon their hydrophobicity or their hydrophilicity. In other words, how hydrophobic or hydrophilic were these molecules. Okay, so the four pigments again that we wanted to separate were chlorophyll B, chlorophyll B, chlorophyll A, xanthophyll, and carotene. So what we're going to do, we're going to say, okay, let's find out which of these is the most polar, which of these is the least polar. Okay, this chlorophyll B is actually the most hydrophilic. Carotene is the most hydrophobic. Hydrophilic molecules love interacting with other hydrophilic molecules. They also can love interacting with polar substances. So this is going to be the most polar. This will be the least in terms of these molecules. So what role does this play in everything? Well, here's what role it plays. We're going to take a piece of paper that's called chromatography paper. And this chromatography paper is fairly polar when placed in combination with a solvent called chromatography solvent. So what we're going to do, and remember this, is we're going to take a test tube. We're going to place this piece of chromatography paper into the test tube. Okay. And what we did was we took a piece of spinach leaf and we scraped it on the chromatography paper. Okay. So this blotch right here technically contains all of those pigments that I just talked about in it. We then place chromatography solvent, a combination of petroleum ether and acetone, into the base of this. And what we see happen is the solvent starts to diffuse up the paper. And it starts to move up the paper all the way to the top if we allow it. As it moves, the pigments inside of this smear will also move along with it. Depending upon their hydrophobicity or hydrophilicity, how hydrophilic or hydrophobic they will move at different rates up the paper. Those that are more hydrophobic are less likely to form interactions with the paper and will therefore move farther up with the solvent front. While those that are hydrophilic will actually interact with the paper more often, so it means that they're binding to the paper and it's impeding their progress. Okay? So, knowing which of those is the most hydrophilic and which of those pigments was the most hydrophobic, think about which pigment you expected to find at the very top and which pigment you expected to find closer to the bottom. Okay? Think about the actual way that this chromatography paper looked when you finished it. You had two lines of these pigments and then a smear here that represented a pigment and a line at the top that represented a pigment. So using the information I've told you here, figure out which pigment you expected to see at the very top of this paper, which pigment you expected to see at the bottom, and where you expected the other ones to wind up as well. Okay? The one that winds up here at the top is going to be considered the most soluble. Okay? Because it technically was the most soluble in our solvent. It moved pretty much with that solvent front. The one down at the bottom, on the other hand, will be the least soluble. So, depending upon how hydrophobic or hydrophilic these pigments are, that will determine how willing they are to interact with something that is polar, like the paper. And that also will determine how soluble they will be in this solvent front as it moves through that paper. Okay, so I've given you all the information. Now use that information to fill in this part here. Okay, tell me where each of those pigments are going to wind up, where you expect to see those pigments.